Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
Linus Pauling, a towering figure in 20th century chemistry and a recipient of two unshared Nobel Prizes, one in chemistry and one for peace, possessed an intellect that ranged from the intricacies of chemical bonding to the complexities of international politics. Among his extensive contributions to science, his 1954 paper, Structure of Proteins, Two Hydrogen-Bonded Helical Configurations of the Polypeptide Chain, stands as a pivotal moment in the history of molecular biology. This work, published in the Proceedings of the National Academy of Sciences, unveiled groundbreaking insights into the three-dimensional architecture of proteins, proposing two helical structures, most prominently the alpha helix, which revolutionized the understanding of protein structure and function. The genesis of Pauling's work on protein structure lies in a rich confluence of factors: his deep understanding of chemical bonding, particularly the hydrogen bond, the burgeoning field of X-ray crystallography, and the growing recognition of the central role proteins play in biological processes. Prior to Pauling's investigations, the structure of proteins remained largely an enigma. While the amino acid composition of proteins had been established, how these amino acids arranged themselves in three-dimensional space to perform their diverse functions was a question that eluded definitive answers. The prevailing model, often referred to as the cyclol theory proposed by Dorothy Rinch, suggested that proteins adopted a globular structure formed by covalent bonds between amino acid residues. However, this model faced increasing challenges, particularly from experimental data that suggested the importance of hydrogen bonds in maintaining protein structure. Pauling's approach to deciphering protein structure was deeply rooted in his understanding of the fundamental principles of chemistry. He recognized the crucial role of the peptide bond, which links amino acids together, and the potential for hydrogen bonds to stabilize specific conformations. Crucially, Pauling insisted upon adherence to known bond lengths and bond angles derived from small molecule crystallography, a principle he termed the rigid body requirement. His insistence on physically plausible models, guided by fundamental chemical principles, distinguished his approach. Furthermore, the availability of X-ray diffraction data, even though limited and often of poor quality at the time, provided crucial experimental constraints for his model building. The 1954 paper detailed two specific helical structures for polypeptide chains stabilized by hydrogen bonds. The first, and most famous, was the alpha helix. In this structure, the polypeptide chain is coiled into a right-handed helix with approximately 3.6 amino acid residues per turn. The helix is stabilized by hydrogen bonds formed between the carbonyl oxygen of one amino acid residue and the amide hydrogen of an amino acid residue 4 positions down the chain. These intramolecular hydrogen bonds run nearly parallel to the helical axis, providing significant stability. The alpha helix, as Pauling astutely suggested, allowed for the maximization of hydrogen bonds while adhering to known bond lengths and angles. He also introduced the gamma helix, though this structure receives less attention. The second structure proposed in the paper was a less compact helix which also utilized hydrogen bonds to maintain stability. While the alpha helix proved to be a ubiquitous structural motif in proteins, the other helical arrangement has not been observed as often. The impact of Pauling's alpha helix model was immediate and profound. It provided, for the first time, a plausible and chemically sound explanation for the secondary structure of proteins. The alpha helix quickly became a cornerstone of structural biology, explaining the repeating patterns observed in fibrous proteins such as keratin and myosin, and providing a framework for understanding the more complex structures of globular proteins. One early application of the alpha helix model involved understanding the structure of alpha-keratin, the protein that makes up hair, wool, and nails. Pauling and his colleagues showed that the X-ray diffraction patterns of alpha-keratin were consistent with a coiled coil structure formed by multiple alpha helices wound around each other. This model elegantly explained the strength and elasticity of these fibrous materials. Furthermore, Pauling's work directly influenced the subsequent determination of the three-dimensional structure of myoglobin by John Kendrew and his team, and hemoglobin by Max Perutz and his group. These monumental achievements, which earned Kendrew and Perutz the Nobel Prize in Chemistry in 1962, relied heavily on Pauling's insights into the importance of the alpha helix and the use of model building guided by X-ray diffraction data. Indeed, Perutz initially struggled to interpret the intricate diffraction patterns of hemoglobin until he was shown a preprint of Pauling's paper. He later credited Pauling's alpha helix with providing the key to unlocking the structure of hemoglobin, famously stating Pauling's alpha helix turned out to be the Rosetta Stone of protein structures. The acceptance of Pauling's model was rapid, though characteristically in science, Not without initial skepticism, some crystallographers, accustomed to the inherent ambiguities in X-ray diffraction data, particularly in the early days of the technique, found it difficult to accept a model based so heavily on theoretical considerations and model building. Dorothy Rinch, a proponent of the cyclone model, remained a staunch critic of Pauling's helical structures. She maintained that covalent bonds, rather than hydrogen bonds, were the primary forces holding proteins together. However, mounting experimental evidence, including high-resolution X-ray structures of proteins, overwhelmingly supported Pauling's subsequent research built elaborately upon Pauling's foundational work. The determination of the structure of beta sheets, another crucial element of protein secondary structure, further solidified the understanding of protein architecture. The development of methods for predicting protein structure from amino acid sequence, while still a formidable challenge, rests on the principles established by Pauling regarding the interplay of chemical bonds and steric constraints. Pauling's 1954 paper also had a profound influence on the development of molecular biology as a discipline, by demonstrating that the structure of a biological macromolecule could be understood in terms of fundamental chemical principles. Pauling helped to bridge the gap between chemistry and biology. This paved the way for the emergence of a new field that sought to explain biological phenomena at the molecular level. The direct influences on Pauling's work can be traced to several key sources. His deep understanding of chemical bonding, cultivated over decades of research, was paramount. His work on the nature of the chemical bond, culminating in his seminal book The Nature of the Chemical Bond, published in 1939, provided the theoretical framework for understanding the forces that govern molecular structure. The X-ray diffraction studies of small molecules, particularly those performed by Linus' contemporaries, such as William Asbury, Rosalind Franklin and Maurice Wilkins, provided crucial experimental data and insights into the dimensions and arrangements of atoms in molecules. The influence of Pauling's work on others is equally clear. As discussed previously, Kendrew and Perutz directly applied his insights to solve the structures of myoglobin and hemoglobin, respectively. James Watson and Francis Crick, in their groundbreaking work on the structure of DNA, were also influenced by Pauling's approach to model building and his emphasis on the importance of hydrogen bonds. Indeed, Pauling was also working on the solution of the structure of DNA and was thought to be a serious competitor to Watson and Crick. He was ultimately delayed in solving the structure, partly due to his erroneous assumption that DNA contained 3 strands, fueled by an incorrect interpretation of certain X-ray diffraction. Nevertheless, Pauling's contributions to understanding molecular structure earned him the moniker of one of the fathers of molecular biology. In conclusion, Linus Pauling's 1954 paper, Structure of Proteins to Hydrogen-Bonded Helical Configurations of the Polypeptide Chain, represents a watershed moment in the history of science. It unveiled the alpha helix, a fundamental building block of protein structure, and established a framework for understanding the intricate 3-dimensional architecture of proteins. Pauling's insistence on adhering to fundamental chemical principles, combined with his astute interpretation of experimental data, revolutionized the field of structural biology and paved the way for the emergence of molecular biology as a distinct discipline. It stands as a testament to the power of chemical intuition and rigorous model building in unraveling the complexities of life at the molecular level, a legacy that continues today. Structural biology has benefited from advances in computation and cryo-electron microscopy. However, the principles of secondary structure first delineated by Pauling persist, serving as a bedrock for decades of structural analysis to come.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.